Yeah, thank you much for the introduction, and thanks for the opportunity to talk. Just one thing for the environmental enrichment, I think was Beneku who did discover the nicotinic channel, and that was more or less due to the fact that they did smoke while they do patch clamping, and they could then, when they were smoking, there was more current there. But I want to talk today about what happens basically here in Qatar with QCRC. We have our laboratory in QSTP, Qatar Science and Technology Park, a nice laboratory, and we will do there the research together with the colleagues here from the clinic. And I will talk about the whole heart, what happens there, the clinical relevance, tissue slices, single cells, and give a little bit of an outlook what we want to do. Of course, these what I present now is more or less ongoing work or work from the past. Going back to the heart, here is a Langdorff, also a heart perfused with solution on a so-called Langdorff apparatus. You see here one atrium with a guinea pig heart, and that is we're going to look at. And you see it is nicely beating, so also outside the body, the heart is beating due to the sinoatrial node. And if the heart is in the body, it is pumping the blood around, which is very essential for our life. And it is dependent basically on the calcium in the cell itself. And the calcium beat by beat undergoes a concentration difference. It is very low calcium in the beginning, and then it goes up. The contractile elements contract and enable the heart to pump something out, and then it goes back to a baseline. And the heart is electrically driven. The sinoatrial node in the right atrium is then spreading around the signal over the heart, and then in this coordinated manner, basically the heart can beat nicely all night long. Why we are interested? Of course, we heard a lot about the malfunction of the heart, and obviously these can kill. What we can measure is, for example, the cardiac output and the force development of the single cell. We can look at the heart rates, and we can look at the sinoatrial node function, and we can see an ECG or a pseudo ECG on an isolated heart, as you could see. We can look at the cell-cell connection. And very important for us is as well the correlation between calcium and voltage. To go voltage and calcium, to look at them, we use, for example, voltage-sensitive dyes or calcium-sensitive dyes. These voltage-sensitive dyes can attach to the membrane, and they are excitable. They are fluorescent dyes, basically. You use a specific wavelength of light to excite them, then they give you a fluorescent signal. And the intensity of the fluorescent signal will be dependent on which membrane voltage you have. You saw the action potentials before, -80 to +40. There's a range when then the intensity is changing. The calcium dye, you try to bring in the cell, so you measure calcium intracellular. And again, you can use different wavelengths, and the intensity is as well correlated to the concentration. Why I'm mentioning here different wavelengths is because we want to use both of the dyes in one preparation, so we need to separate basically the voltage signal and the calcium signal from each other. How does it look like? So here you have a cardiac preparation. Here you have, for example, a stimulation point, and here we measure, and you can see the upstroke of the action potential. And this is an intensity plot basically of the dye, so the baseline is here, and then with changes in the action potential, the intensity of the dye is increasing and going down, and it's nicely correlated to the dye. A lot of studies have been done that this is really the same reproduction of the signal, similar to the patch clamp technique. Also for the calcium, you have here a baseline, and if the cell gets excited, the calcium goes up and goes down again. So they removed out of the cytosol. Here is an intensity plot, basically. We stimulate here the cardiac preparation, and then it's spreading around. You see, it gets brighter. and is then fully excited here in the end. And in the calcium signal is a little bit delayed. The peak of the calcium signal is delayed to the peak of the voltage signal and is following whatever the voltage is telling it. How does it look in a movie? Very nice. You saw here the atrium, and then it's spreading around the whole heart. This is a voltage signal on that side, and the calcium signal on the other side. And this is now followed by a little slower motion. So here the atrium is excited. Excitation is very quickly around the heart. This is a red heart actually. And then we stimulate here on that point with a point stimulator, just to make the point, that you can influence as well the whole system. You don't, necessarily have to work with this atrium. You can stimulate the heart as you wish, basically, and apply protocols. And if you apply a protocol where you can bring also the heart into fibrillation, you can follow this fibrillation pattern, and I think that's very nicely to look at. And you have here, something is going around here. You have so-called singularity points, you have breakthrough points, and there you can do a whole lot of studies with these images. And we hope to commence these studies as well very soon here in Qatar. And we cannot only look at it for 2 seconds, 3 seconds, or 4 seconds. With the technology nowadays, it's possible to look over it for minutes or even a half an hour, and we can then extract the information from the voltage and from the calcium and analyze it over a long stretch of time. So we can look, we can provoke a fibrillation, we can add drugs, we can follow what happens there, and especially interesting the transient periods between the two states. If you don't want to work with the whole heart, we can slice off a piece of the left ventricle. So it's you prepare the left ventricle and then you slice a thin slice. This is maybe 300, 400 micrometer thick, and it's stimulated here. So it's a stimulator here, and that's why it's looks like maybe a little moving something. This is of course a slice basically which is very alive and which is showing that there are a lot of living cells in there, and we can use these preparations very nicely. The good thing is with slice preparations out of one heart you can have 10 to 12 slices. You can do different preparations, different drug application, different studies on it. And then you have them here, you restrict the motion, and then you can stimulate them here. You see how nice the voltage travels through the slice, and also the calcium follows up. This is a red slice again. And then you can imagine in preparations like that, we can add a drug. How does the drug change the action potential or the calcium signal? And since both are intercorrelated with each other, that gives us some insight into the function of the heart. Going down to the one more level, to the single cell, this is a very simplified picture. We saw as well some nicer membrane pictures with the protein in there, and the proteins can be quite a lot of diversity of proteins in the heart cell, obviously, but also the channels. And we can look at these channels with patch pipettes, so with a patch clamp method. So you have a glass pipette which you attach to the cell, and then you can control what is the membrane voltage, and you can measure as well the current over this membrane. And then you can measure, for example, an action potential which is produced by one cardiac cell. Or you rip off this piece of membrane, then it sticks to your pipette, and you can measure the current only over this little piece of membrane, which is of advantage if you only want to look at a few of these proteins, not at all at the proteins in the cell. How does it look like? So here we have, for example, a patch pipette. We have here then what my colleague Jens already mentioned, the possibility to attach two little sticks. It's called carbon fibers in that case, where we restrict the movement of the cell towards this side. So this is this will be restricted in motion, And here we attach a second carbon fiber, which enables us to stretch the cell, to stretch the area between this carbon fiber and this carbon fiber. And so we can study, for example, the mechanical effects onto cardiac cells, measure at the same time the action potential, for example, and a protocol like that could look in that way that we have, normal action potentials. We look at them, and for only one action potential, we stretch the cell and then see whether there's a difference in that action potential or not. And how does this change if you have, for example, some stretch situations, additional drugs, or also cells with eventually mutations of different channels. We can also look at the current through a particular channel. So this is the ATP-sensitive potassium channel. In a normal cell, there's nearly no current going through that channel, but if you reduce ATP in that cell, then you provoke huge current through that channels, and that is basically then the baseline. If we apply stretch to that situation, then you can see we can even increase the current that is going through that channel. If you release the stretch here, then it goes back to the baseline. So that's a very interesting mechanism, which you will be able as well to reproduce here. If you look for a few channels only, then we can look at the a single cell current basically here in that case. That's the control case, and then we add, for example, a blocker, in that case IPER is 2 micromolar, and then the open probability of that channel is decreasing. That was a study where basically we used amiloride, diclofenac, and IPER. Not only in combination with patch clamp, but also if you want to look at the calcium sensitivity, we can look at skinned myocytes. So these myocytes basically attach as well to two pieces where we can stretch them, from one length to another length, and we can study very nicely the calcium dependence of their force production between different sarcomere length differences. And that will be very interesting, especially for mutation of the, for example, myosin binding protein C. So here I also come already to the future work as QCRC to mention but a few, I think, not everything. We, of course, are interested in the Bugarra syndrome, in inherited cardiomyopathy, especially the myosin binding protein C. And we will look forward to use animal models to look at fibrillation, mechanical impacts, drug sensitivity. And what we want to do is as well to integrate all levels, single cells, slices, the whole organ, and then we have a better picture of what happens basically in the cell. We are very dedicated to the basic science there basically, and beside the electrophysiology, which I'm talking here now, we also do stem cell research, biomarkers, calcium signaling, molecular structures, and a few more things. You heard yesterday about the zebrafish model, for example. And that means we are very multidisciplinary, and we hope basically by our approach that we can talk to each other, we can communicate with each other, talk about different possibilities, what we can do, and give it a chance as well, that we discover as well something by chance, and maybe we will once have a very great breakthrough due to our approach here in an interdisciplinary team. I want to thank Samek Yacoub and the QCRC team for the electrophysiology, Yunus, Abu Matthias, and Rasik. Work involved basically in the pictures you saw, thanks to Peter Kenmaya, Andrew, and Peter Cole at Imperial College, Oxford University in Madrid, and thank you for your attention.
If I may start with one. In addition to the genetically modified animal models from which you could isolate cells to perform these experiments, would it also be possible to apply them on cell cultures where you have cells transfected with the mutations that one may be interested to study?
Yes, yes. So we are looking forward. Basically, we see a stem cell team, which is getting patient material, mutations, and produce cardiac stem cells basically, and then derive them into maturation that they look like cardiac myocytes. There in the cell culture dish, and we hope. So we did already preliminary work to that, so we can use our voltage-sensitive dye, our calcium dyes, and we can look at them in the dish. Yes. And we're looking very much forward to that. Yes.
Well, I would like to congratulate you and the team for this setting that you have and all these kinds of possibilities that you are. My question was a little bit on the modified genetically modified animals. Can you elaborate a little bit on that, on which type of models you have to genetically modified animals, and how can you?
Okay. So. This, in terms of genetic modification, we hope basically over the Bugada project or inherited cardiomyopathy project that we will have mutations, and either we have to bring them to introduce them into commercially available iPS cells, which can derive into cardiomyocytes, or if we are kind of in the situation to obtain tissue, skin tissue, also to develop stem cell lines, basically, that would be great. That would be the greatest opportunity. And then that is basically on cell culture level. We hope to work as well with specific mutation we will introduce into mice models or the zebrafish model. And there we can as well stain the membrane and look at the signals. Yeah.
In addition, we have access to human tissue from defined patients with defined mutations, particularly with HCM, where we have more than 100 patients undergoing myectomy. Almost we can do 7 in a week. And we know that they're carrying myosin binding protein C in the following. Combined with what you were talking about, Despina, having IPS from these patients and then differentiating it with the cardiomyocyte. Christopher is here and the team who are actually almost regularly now showing IPS differentiated into cardiomyocytes, and you can see them beating. They're still not carrying the mutation, but that's the next step. It could be applied to humans quite rapidly.
Thank you. If there are no other questions, yes?
Is there anything known about mutations causing the disease and altered stretch response?
I see. Okay, the stretch response is one of the methods we can use here. Of course, we can look for other things like pH or other environmental factors. That's without any question. The stretch is very interesting for the inherited cardiomyopathy. The changing in myosin binding protein C, though the binding affinity to calcium is different, the binding is different, the force development of the cell is different, and there it becomes very interesting how that interferes. Is more calcium available, is less calcium available? Would more calcium availability maybe heal or cure the disease? And that's
it shouldn't be very important because during the action potential, the heart cells contract to become shorter, then it's stretchable.
No, not during the contraction, the cells first get pre-stretched, and then they contract again, start to their maybe original length. And there are studies already which show, I think, if you have cells and you clamp them on the length, then you have a different calcium signal than instead to let them freely move. So there are mechanical influences of the microskeleton, which is interfering with the CAM kinase 2, with the ryanodine receptor and other things. And if that is disturbed, then you have also their problems.